So good morning. My name is Eric Halioua and I'm President and CEO of PDC Line Pharma. I'm going to present you over the next 15 minutes the activity of our company, PDC Line, which is developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. The company is a spin-off from the French Blood Bank, and the scientific founder of the company, Joël Plumat, when he was in an academic research lab associated to the French Blood Bank, has identified in the blood of a leukemia patients a very rare leukemia affecting the plasmacytoid dendritic cells, which is the smaller fractions of the dendritic, main fraction being the myeloid ones. And the leukemia was affecting these small fractions. And based on the blood of his patient, he developed a GMP cell line and a master cell bank GMP approved for clinical use that we are still using today as a vaccinal platform. We have a team of more than 25 people, including seasoned executives from biotech and pharma. In terms of funding, we get close to 32 million euros so far, with a part in non-dilutive money. Our technology, or PDC cell line, has three key advantages. First of it is a cell line, so it's extremely easy to expand. It's an immortalized cell line. The origin of the cells, being a plasmacytoid dendritic cells, provide very interesting biological property, and it's highly potent antigen-presenting cell. And last but not least, the platform is extremely versatile. In fact, we just have to load the cell line with peptide representative of the tumor antigens, and it's broadly applicable to any kind of cancer. We are clinical stage. We have already completed a phase 1b academic study in melanoma, and we have a phase 1/2 ongoing in non-small cell lung cancer. The pipeline is summarized here in this slide. We have three components. The first one, PDC-MEL, the melanoma, with the 4 shared tumor antigens, as I said earlier, has been completed. The result has been published, and the study is not active anymore. We are today focusing on the 2 other components, the PDC lung, with 6 shared antigens, which is ongoing in the phase 1/2, and the PDC neo, which is this time using neoantigens, which are shared neoantigens, which are antigens related to the mutations induced in the tumor. Important element to mention, it's that we have signed already our first licensing deal. It was at preclinical stage. In early 2019, we signed a licensing agreement with LG Chem to develop PDC lung cancer vaccine in Asia. LG Chem is a very large organization. LG is a very big conglomerate, but LG Chem is the 10th chemical industry in the world. And we signed a license with the life science company of LG Chem. We provided exclusive license in South Korea and options for the rest of Asia. And the total deal value is $123 million, plus significant tied royalties on net sales in Asia. One of the key assets of the company is definitely the management team. We have very experienced and competent people in the team. For example, Shanna De Bruyne, that you see in the bottom left, our medical director, is coming from GSK Vaccine. She led all the cancer vaccine, MAGE A3 of GSK in lung and melanoma. We have as well Claude Dedry, the VP of Pharmaceutical Operation and Quality, in charge of the CMC in the company, that has a very extensive experience in cell and gene therapy. We have Laurent Lévy and Joël Plumat that have co-founded with me the company. So Joël Plumat, the scientific founder, joined full-time the company. He's an immunologist and the co-inventor of the technology. Laurent Lévy is now the CFO, CEO of the company, COO, sorry, of the company, in charge of finance. And myself, Eric Halioua, I've been, as we said today, a serial entrepreneur. I've co-founded several biotech, like Musics, Murigenetics, and board member of different companies. I've been involved in several M&A or IPO in the past. We have as well a very good governance, a very strong board of directors. We have as a president of the board, Monder Majoubi, who is an oncologist and the current CEO of Innate Pharma, but he was as well the senior VP in charge of oncology of AstraZeneca, of Genentech and others. We have as well Jean-Paul Priels, who is the former global R&D director of GSK Vaccine, but is a board member and investor in the company. We have Alain Herrera, that you see on the bottom of this slide, which is an oncologist as well, and the former lead of the business unit oncology of Sanofi. We have as well representative of the investors like Sang-Woo Lee, who is the head of a Korea investment partner, which is one of the biggest private equity and VC fund in Asia. He's based in US, but is one of the board member of the company with representative of others investors. The company made significant achievement over the last 24 months. I'm going to mention them quickly. In January 2019, we get the Gmp Accreditations by the Belgium agency for the QC and release activity of our vaccine PDC lung. We signed in March 2019 the licensing deal with LG Chem. We get the authorization to launch the phase 1/2 with our cancer vaccine in lung cancer in Belgium and France in June. Our medical director, Shana, arrived in August 2019. In December, we closed a 20 million B round of financing led by the multi-million Asian VC Korea investment partner. We announced in February 2020 administrations of a first patient with the cancer vaccine. In June, we get the arrival 2020 of Monder Majoubi, the president of the board, and in September, we announced the first dose patient with high dosing cancer vaccine candidate. Since then, we get the approval as well in Germany to initiate the clinical trial. So just a few words about the technology, how it works. We have a master cell bank, GMP approved, where in bags we have the cell line coming from the blood of the leukemia patients that are stored in liquid nitrogens. We have developed several working cell bank from this master cell bank, and we just have to take one bag of this working cell bank to seed a single-use bioreactor. The cells, after 9 to 10 days, multiply suspensions in a synthetic medium without any, it's a serum-free, without any required GMP interleukin or whatever. We just have to grow the cells, then harvest the cells, to split the cells according to the number of GMP peptide we want to load on the HLA present in the membrane of the cell line. We load them individually, then we pull the cells and irradiate to stop the proliferations. Then we put the cells in a specific container vial. It's the technology of aseptic technology, where the cells are put with a cryoprotectant. It's stored and it can be stored in liquid nitrogen afterwards. Then we ship the cells to the hospitals in minus 70 or 80 degrees fridge. And when the patient is ready, we just have to thaw the cells and with a syringe to take the cells and infuse to the patient. So it's extremely easy process, very scalable. The batch-to-batch consistency is extremely high and good. In this case, the raw material is exactly the same. The cell line is exactly the same all the time. And the cost of good as well is extremely low compared to the other approach. The second key advantage of our technology is the potency. We did different benchmark study to compare our PDC line loaded with a specific antigens, which is Melanoma, with other classical approach, which are the dendritic cells coming differentiated from monocyte of patients that are in general myeloid DC, autologous or allogenic DC. And as you can see here, by FACS dextramer, we assessed after 21 days of culture in vitro the capacity of the cell line to expand CD8 cytotoxic T cells specific of these antigens of Melanoma. And as you can observe here, we are 20 to 200-fold more potent than the other approach. We did as well study with neoepitope, neoantigens, which is a hot topic currently. And for that, we have produce mutated peptide where the mutations you see in red has been clearly established, and these neoepitope were identified, characterized, and published in the literature, scientific literature, in the blood of melanoma and lung cancer patients. Then we loaded our cell line with this neoepitope and put in presence of the purified CD8 T cells of 3 healthy donors. What you see here, HD01, HD02, and HD03. As you can see here, after 21 days of cultures, in red and purple, you can see by fax the expansions of the CD8 T cells specific of each of these neoepitope. We have more than 150-fold increase in terms of CD8 T cell specific of antigens. So that is extremely good result, knowing that in healthy donor where all the CD8 are naive, the precursor, the prevalence of this precursor is less than 1 out of 1 million. In terms of IP, we have 3 key pillars to protect our technology. First of it, we have our proprietary cell line, which is unique, and it's come from 1 patient, which is dead now. It has been licensed from the French Blood Bank, and the master cell bank is clinical grade and fully characterized and approved. We have as well patents, 2 family of patents protecting the cell line and its applications has been granted already. And we filed more recently a new family of patents related to the genetic optimizations of PDC line. Third pillar is all the know-how and data related to the manufacturing process, immunomonitoring assays, and the preclinical data. Okay, just a few words about the melanoma clinical trials we have completed. This study was done in France on 9 patients with advanced melanoma, phase 3 or phase 4. The vaccine was cell line loaded with 4 different antigens, tyrosinase, GP100, melane, and MAGE-A3. The patient was infused 3 times, and the results were definitely very promising. We have observed acceptable tolerability and safety profile. That was the primary objective. We didn't observe any allogenic rejection detected through antibody against the mismatch HLA of the cell line in any of the patients. And we have observed as well significant antigen-specific CD8 T cell response in the blood of the patient after infusing the vaccine that were directly detectable in the blood, so without any in vitro expansion. So showing that the vaccine can be biologically active in this setting. And that despite we infuse just 3 times, and despite we didn't co-administer any checkpoint inhibitors. And we know there is synergy between our vaccine and checkpoint inhibitors that has been demonstrated in this study. We have collected the blood of melanoma patients, and you know melanoma is a very hot tumor with a very high tumor-induced immunosuppressions, and we loaded the cell line with melane, which is an antigens, still the same antigens, and put in culture with the PBMC of melanoma patients. And as you can see here, after 1 week of cultures, we have 15-fold increase with the PDC the vaccine alone. If you put the checkpoint inhibitor alone, anti-PD-1, we have almost nothing. It's multiplied by 2 compared to baseline in terms of expansions. But if you combine both, is where you see this nice synergetic effect where we have more than 35-fold increase in terms of CD8 T cells specific of the melanie. But this result has been published as well. It has been done in a cohort of 13 patients to demonstrate the synergetic effects. Okay, let's pass now to the clinical trial, which is ongoing now on non-small cell lung cancer. You see here the 6 antigens we have selected for the lung vaccine. There is MAGE-A4, MAGE-A3, MUC1, New York is 1 surviving, and multi-MAGE. And we have for all of these antigens in the blood of healthy donor demonstrated that we can expand CD8 T cells specific of each of these antigens. You see here baseline and post expansions, the expansions of the clone of CD8 T cells specific of each of these antigens. And as you can see, it's quite extensive for all of them after 2 weeks of culture. The design of the clinical trial is presented in this slide. The objective is to demonstrate the safety, the immunological activity, and clinical response of our vaccine, PDC lung, in association with checkpoint inhibitor. And we have 4 cohorts of patients. On the 64 patients we need to enroll. The 2 first cohorts are going to dose to test 2 doses of vaccine, low dose and high dose, on patients with on adjuvant setting on monotherapy with the vaccine alone. So we have 6 patients low dose monotherapy. This cohort is completed already. Then we switch to the high dose monotherapy. And in parallel, we switch to the second populations, which is the one where we would like to observe the clinical benefit. It's first line in combo with first line metastatic non-small cell lung cancer in associations with checkpoint inhibitor. First with low dose 6 patients, then we can go to the high dose in combo with checkpoint on 40 patients. We will administrate IV and subcut every week. So 6 infusions will be done. That is the administration schedule. As I said, we will test 2 doses, 2 million cells per peptide and 12 million cells per peptide. To finalize my presentations, the funding strategy and the timeline, we get a B round of financing of 20 million euros end of 2019. With this money, we would like to complete the current clinical trial in lung cancer, and we hope to get the final report Q1, Q2 of 2023. We expect to have the interim report of the clinical trial mid-2022. And based on this data, to launch a new round of financing, an IPO or a global licensing deal. And today we are open to these different options. And with this money to finance a second clinical trial, a randomized, that will be a proof of concept, and a new phase 1/2 in over indications and setting up in US and Asia. On that, I would like to thank you for your attention with a nice photo of the team. Thank you.